Good morning. This is Joanne Geer. I'm the Executive Director of the Biopharma Research Council, and I want to welcome you to our webinar today. We're just going to wait just a few seconds to let a few more people get themselves logged in, and then we'll begin. Well, hello there. I want to welcome everyone today. I am the Biopharma Research Council hosts a number of webinars throughout the year. We are an association for scientists throughout the biomedical research community, which means industry, academia, nonprofit, government, supplier labs, and their teams. We run multiple webinars, we run conferences several times a year, and our best ideas come from our community. So if today's program gives you any ideas, please feel free to be in touch, because we may well be able to build something that intrigues you specifically. This morning we're going to hear on a program on optimizing preclinical imaging workflows with online tools with James Denegre from the Jackson Lab and Tim Colbago from imagequality.com. I'm just going to share with you a few upcoming programs. Next week on Thursday at 1 p.m. Eastern, we're having a really interesting program on advancing tissue imaging. And this is a very focused functional and morphological extracellular matrix characterization with stain-free non-linear microscopy. And I've seen the slides. It's a very interesting program. Coming up in the following week in Frederick, Maryland at the National Cancer Institute, we're doing a free one-day conference on altering the microbiome, can it impact health? This is a follow-up from a series we did last spring, a short course in the microbiome, 4 different webinars which are all available through our website. And then the other one I want to highlight is that we're coming to Durham, North Carolina for the 4th time to do the Triangle Biotech Research Symposium, which is focusing on technology convergence this year. We'll be looking at CRISPR, we'll be looking at tissue on chips, we'll be looking at the overview of how to manage technology convergence as it relates to building products and companies and therapies. You should feel free to enter your questions at any time. If you're having any issues, I'll be happy to try to address them, but your questions about the talk are very valuable to us. And if it's about a specific slide, if you'll note which slide when we come to the questions after the presentation, then it'll help us to answer them for you. And I'm going to turn things over now to Tim Colbago.
All right. Thanks, Joanne. Thanks very much, and thanks to the Biopharm Research Council for hosting Jim and I today. It's super awesome. I think last time we got to see each other, I was at one of your shows, and we were talking about Google Glass. Actually, that was a while back, but that was a whole lot of fun. So everyone, welcome to the webinar today. I'm Tim Colbago. Just by way of quick introductions, I'm the CEO here at ImageIQ. And we have built one of the online tools that Jim's going to be telling you a little bit about today that they're using at Jackson Lab called imagequantify.com. Now, I thought we would start with a little bit about Jim, and so you know who's going to be doing most of the talking, because he's going to give you the meat of a lot of this stuff. Jim's an accomplished research scientist and a manager at the Jackson Laboratory, and we've had the opportunity to visit him up in the Northeast U.S. there, and it's been a great time. You know, Jackson Lab's an independent nonprofit biomedical research institution, and it's actually a National Cancer Institute designated cancer center. And Jim's been so involved in all sorts of different kinds of imaging, image analysis, microscopy, digital pathology, flow cytometry, you name it. Jim's got quite a bit of experience on it, and he's given you know, a lot of presentations in France and in MIT. He's been contributor to like well over a dozen peer-reviewed papers. So with Jim here, thanks Jim for joining us as well.
Thanks,
really appreciate it. And what we thought we would do is start the webinar with just a little bit of a challenge that we have seen collectively, you know, Jim and I, and I'm sure a lot of you have seen in this process of of imaging in preclinical research, right? One of the things that we're discovering is starting to come to the forefront when you're acquiring images in preclinical research is this concept of bias and reproducibility that is starting to really start to have a spotlight shown on it. You know, there is inherent bias in manual analysis, and one of the challenges that we have seen in the scientific community is how can we remove biases, whether we know that they're there or whether we know we're not there. And you know, it's interesting to point out that non-blinded observers show to overrepresent predicted results by 27%. Which is, you know, pretty significant, and that's a big thing. But recently there was a really interesting study done on NIH funding here in the U.S., and that roughly $28 billion with a B, $28 billion of preclinical research is not reproducible. And certainly, and you know, Jim, you and I've talked about this ad nauseum at times, right? You know, one of the pillars of good science is the ability to describe what you've done and have someone else reproduce it. And so to sit back and say, hey, look, if I'm doing imaging and I can't reproduce my results, what are we doing? Are we wasting money? Is something else going on? What do we do? So those are some of the challenges that we face when we do imaging in preclinical research. And so what we want to do is talk a little bit about how Jim and the Jackson Lab have overcome some of those challenges. He's going to talk to you about the COMP and IMPC organizations, what those projects look like, the scope of those projects, and go through a couple of case studies with you. We're going to talk about some Faxitron stuff, which is an X-ray work that they're doing in imaging. We're going to talk about OCT layer analysis, interesting, which is, if you haven't seen OTC, optical coherence tomography, really interesting stuff in a newer modality, and some fundus analysis of the eye, which is a little bit less slices and more straight-on imaging. And then we'll talk a little bit about their solutions. And I'm going to spend a little time later at the end of this just talking about the technology enablers that you all have available to yourself and why some of this online stuff can actually happen now. So with that, I will let Jim talk about the interesting stuff of how he's solving some of the imaging challenges in the Knockout Mouse project. Jim, it's all you.
Thank you, Tim, and thanks for inviting me. Here at the Jackson Lab, we're involved in a large international collaborative effort to make a knockout mouse for every gene in the genome. And what that means is we target all the genes that have, the entire genome has been sequenced, and so the identification of all the genes has been already made. What's not known is what do all of these genes do. There's certainly function associated with many genes, but there's not a comprehensive database or a comprehensive knowledge of what all these genes are. So the Knockout Mouse project is designed to make a knockout mouse, in other words, break the gene, and then create animals and ask what's wrong with these animals, to phenotype them. And by understanding what deficits are in the animals, you can work backwards and say, well, this gene is involved in hearing, or in vision, or in motor coordination. This is, it's the first project of its kind in terms of size and comprehensiveness. We call it functionalizing the genome, and it utilizes knockout strains which are the same across all of the centers working on it. So to address Tim's point about reproducibility, the different inbred strains of mice will have very different behaviors in knockout models based on the epigenetics of their genome and the allele distribution. So this provides a standardized set of knockouts and a standardized approach to make the knockout. And so it provides a more comprehensive and reproducible approach to understanding gene function than what the traditional method of an individual lab or PI making a knockout for their own interest will provide. And then, more importantly, the knockout data, the phenotyping, the constructs, etc., are all provided publicly in a large searchable database, which is housed by the Data Coordination Center, which is part of the Knockout Mouse project. May I have the next slide? So in doing this, we're essentially finishing the map of the world, sort of like Columbus did when he sailed off across the Atlantic. And the implications are that these findings are searchable, accessible, and the value of the functionalization is that it can provide direction for translational research, in that there's a large overlap of genetics between mice and humans. So imagine that you've got a disease that has a phenotype that you've never seen before. You can go to the mouse database and look for that same type of phenotype and find genes that are associated with it, and use that to define areas that I might be interested in for human genetics. So the mechanics are that these are targeted knockout alleles, that the phenotyping is done across a number of centers in a collaborative effort, so that the amount of work is spread across and that the pipeline is homogenized in terms of the tests. And on the right side of your screen, you can see an outline of what the phenotyping pipeline is. We've included in it embryo lethality screening, so approximately 30% of these knockouts are going to result in embryonic lethal stages, and this is not completely surprising. If you knock out a standard housekeeping gene, you're not going to make a cell, you're not going to make an embryo, you're not going to make a pup. But out of those embryonic lethal knockouts, a lot of them will have a lot of direct construct towards birth defects and congenital defects. So there's a lot of value in looking at those genes, as well as what happens to an adult when a gene is knocked out down here. And you can see the progress through the pipeline. There are some behavior tests, there are metabolic tests, there are sensory tests, there are blood chemistry tests, eye morphology, vision tests, and a lot of clinical blood chemistries, as well as flow cytometry analysis of spleen tissue and their derivatives. In addition, we're collecting tissues from the adults for gross morphology and pathology.
And Jim, this is Tim, real quick. Each one of these tests, as we see them, I love the color coding on this, right? Because every site is doing some subset of these tests. There's mandatory tests and non-mandatory tests, right? So you're trying to, the goal of this group is to try to coordinate and make consistent all of this tests across, dozens of sites, right?
That's correct. So as you see in the key there, the mandatory tests and the non-mandatory tests are illustrated in colors. And then there's some tests in development, which is a constant process trying to fine-tune the pipeline and get more information. So for the mandatory tests, every center that is doing this is asked to provide these tests with the exact same SOP. And developing these tests in the SOPs was a very long and collaborative effort between all of the centers through large group meetings, both in Europe and in the United States, as well as some very extensive conference calls over time, where all the scientists sat together and sort of put aside their bias about what they liked in a certain type of test or whatever, and came up with what would be the best test to get the best information on a knockout across all the centers, trying to use equipment that was the same or as similar as possible. The reason that there are non-mandatory tests, we like to call it center-specific tests, is that many of these centers have principal investigators who need to have some buy-in onto the program, and so it was decided to allow other tests that were center-specific to enter the pipeline so that the PIs of that lab will be able to generate extra value from these knockout mice as opposed to just the mandatory tests. And then all of these data get sent to the Data Coordination Center, which is in the UK, and they're automatically analyzed statistically and outliers identified automatically and flagged in a searchable manner on a public website. So anyone can get to these information and download the actual test data itself, as well as use the onboard statistics. The next slide. Yes, thanks. So this is a very large project. There are 3 centers at the in the U.S. who are funded by the NIH, and they are centered at the Jackson Laboratory, UC Davis, and Baylor College of Medicine. And then there are 18 international centers. The IMPC stands for the International Mouse Phenotyping Consortium, and that's the umbrella group for all of the centers, including the NIH-funded comp centers. So it's an international collaborative effort. I like to call it the Hadron Collider of biology. It's as large as that physics project in terms of international scope. It's not as large as that project in terms of funding, but we're getting there. So the 3 NIH comp centers are scheduled to create and phenotype approximately 850 to 70 strains of mice in 5 years, and we're coming up on the end of that 5-year period. And the NIH is looking to renew the project, and that's under consideration. There's been a lot of discussion about fine-tuning of the pipeline, review of tests as in which are the most informative, which are the least informative, are there tests for which we're getting multiple hits across multiple paradigms, so perhaps we can dispense with one of the paradigms. So there are 25,000 genes in the genome approximately, and for a full comprehensive analysis that would require 25,000 knockout strains of mice, and we'll need to generate somewhere between 14 and 16 adults per strain, males and females, to get a statistically relevant sampling of what the phenotypes would be. And so this is a large number of animals over a large period of time, and there are hundreds of measurements per mouse for the phenotyping pipeline. So there's an enormous amount of data to handle, and that's an enormous amount of data to collect, to do quality control on. We run wild-type mice through the pipeline in an ad hoc and unknown manner, in a blinded manner, and those control mice test for the QC of the instrumentation as well as users, and provide background data for phenotyping. And so you know, it's an enormous amount of data to collect, to do QC on, to transfer to the central data coordination center, do statistics there, and then present in a very usable manner to the public. And so it's a real challenge to handle this much data. And among those measurements, images are prevalent, and these are among the most difficult to manage in terms of getting their readout. So for things like blood chemistry tests or for behavior tests, the readouts are unequivocal. It's how many times did the animal put its nose in a hole, how many red blood cells are there, etc. These are measured directly. The metrics are standard. The statistics are necessary on that, but in terms of collecting the data and analyzing it, it's pretty straightforward. Images need to be annotated. Somebody has to look at that image and assess if there is an outlier from the expected result of the image. And the most problematic bottleneck in all data, image or otherwise, on the Knockout Mouse project has been X-ray analysis of skeleton. So every center does this test, and there are 5 X-rays created for each mouse, and there are 8 mice which are X-rayed, 4 females, 4 males for each line. And there are 68 parameters measured on these X-rays, and those parameters are things like quantifying the numbers of digits, the number of vertebrae, measuring lengths of bones, and looking at abnormalities in the overall shape of things. If you could go back one slide. Right. So what you see here are examples of the 5 standard presentations of the animal. They come out as DICOM images. And now go ahead one, thanks. Yeah, they're quantitative, and then there are qualitative aspects to this. And currently, every center has an expert who is a skeleton or bone expert, typically a PhD, who is analyzing and annotating these images manually. They take the full data set, open them up, start reading them, make notes, make measurements, enter those into either a spreadsheet or directly into a LIMS system, and then eventually upload those to the Data Coordination Center. This is the worst bottleneck. It's the greatest backlog in terms of annotation. Collecting the images is not particularly difficult. We can upload the data themselves, just the images, and present them, and we're doing that. They don't have to be annotated, but that takes the value away of that image from the searchable format of the database. So if you're interested in skeletal defects, you are absolutely free to look at all these data yourself and analyze them, but it's much more valuable to you to have someone who has some knowledge of it to just flag right away that there's a problem in the skull, there's a problem in the tail, there's missing these number of digits, and it'll draw you right to the outliers instead of you having to screen all of them. So it was really critical to develop some methods to reduce this backlog. And so, and that's why we engaged Image IQ. So the manual analysis is simply not high throughput enough. We're looking at about 10,000 mice in a 5-year period. In addition, when you're sitting there looking at all these day by day or hour by hour, you are going to make errors. It's user error due to fatigue, mix-up of animal IDs, that glossed over eye feeling you get when you're doing the same thing over and over again. And
yeah, Jim, I got to tell you, I went through that. You know, we were doing some validation for some of this work, and I said, I want to see what this is like, and I jumped in. And I'm like, I'll do some of this, right? And man, you do this for, I mean, and look, I probably got a little bit of adult ADD going on, but I mean, you do this for a couple hours, right? And then all of a sudden you're like, that's close enough. Yeah, that's close enough. You know, and you're trying. You know, I mean, like I'm trying to do good validation. And you're like, you know, that glassy-eyed feeling is, you know, it's tough. And so, you know, not only is it an efficiency thing, because at some point you got to get up, walk around, focus on something, not the computer screen. It's also that bias that happens just naturally. It's people stuff, right?
Right. And the last point on this slide is that this manual annotation really drains your phenotyper resources, your expert phenotyper resources, who might be much better, more productive doing their own research. And also, not every center has a true skeleton expert, so they're lacking some of this phenotyping expertise. So the next slide. So we engaged Image IQ to develop some methods for automating the X-ray analysis, and we limited it to some of the quantifiable metrics that that are required, and mostly because that was the simple pilot for this project, which we then presented to the IMPC and to the NIH to their great approval. And the process is here on this slide. We house our images in Omero, which is a free open source image server software with a lot of flexibility, and through that feed the data right to Image IQ through their Image Quantify, I don't know what to call it, their Image Quantify system,
Sure.
And which is cloud-based, and they've developed some algorithms to make the measurements, which we're going to discuss in a minute. Overlay the images, meaning that the measurements are traceable, visual to you. You don't just get a spreadsheet. You see the data, you see what the measurement was, and you can QC it or review it yourself, which is really critical to this process. And then puts out the methodation to validation reports on all the measurements, and then gives you the quantified metrics, which can then be uploaded directly in the format as you see below, right into the Data Coordination Center. Did you want to say something on that, Tim? Or?
Yeah, no. I mean, I think that the interesting part about that whole story is that, you know, the talk we're here is, you know, the online clinical, you know, the online, using this in online preclinical research, and taking something like the Omero platform, right, and using that for what it's really good at is wonderful, because, you know, I love your map, right, because there's so many people touching so many different parts of this project, but that all have to be coordinated. So now you've got this ability in an online way to share that data, to not worry about double entry or anything like this. And when you're talking about the numbers you're talking about, right? I mean, just JAX alone, 10,000 mice, 2,000 a year, making 68 measurements. There's a lot of data that you're collecting, and so you bring that online tool together, and it's not just like you're doing Dropbox stuff, right? I mean, you actually have a place to put this. You get it to the data coordination center, and it's actually orchestrated in a way that makes sense, right? And so I think that's kind of the cool part about that whole picture, right? I mean, it's neat that the image quantify stuff is there, but you've built this pipeline for image analysis that actually allows you to get the data done once, get it done right, and get it coordinated.
Right, and let me add to that. This does not require any center, any user to have on their own desktop to download their own software. So this can be delivered anywhere around the world and to any of the comp centers. Even as you're sitting in an airport waiting for your flight, you can start uploading data and work on it, and it's infinitely scalable. So the context and the approach of putting it into a cloud online is what allows that freedom to provide it anywhere when you need it, and that's a real key for us for the IMPC especially, where it's
for sure. And well, it's kind of like we were talking about before the webinar started when we were in the little green room, right? The virtual green room for the webinar. And I just switched to a Mac. Well, I don't have to worry about any Omero or anything else running on one version or another, right? Of I'm on a Windows machine, I'm on this, I'm on that. You have that access, so that's pretty cool. So.
Yep, great. And the next slide. So here's the case analysis for some of the simple metrics, and that is to count the number of digits. And here on the left, you see the DICOM with the right forepaw, and the algorithm very briefly makes a mask of the image, and then down here it turns it into a single pixel skeletonized version of that, and then finds the endpoints right here and puts a red dot on that, and then that whole thing gets superimposed over the original DICOM. It's a red flag, tells you here are the digits, and it counts them. It says here are 5 digits. So if a digit is missing, it'll say 4 or 3, whatever, and you will be looking at the image with the red dot, and you'll know what it is counting. There may be a problem. For example, the digit might have been folded under, and the algorithm might not have caught that, and you can edit that. Or you can start and note right away, because you have the data on the original image, that, gee, it's always the right side, or it's always digit 1 that's missing, or digit 2. And so there may be more information in terms of its position that's missing from that kind of information. So this allows you to very quickly QC and assess the value of those data. You were going to say something?
Jim, I think that's a good point. One of the things that people get hung up on, and I think even within the Jackson Lab, you guys have you talk about this in a different way. From our perspective, we talk about automated image analysis, but the reality of it is what you're doing is taking out some of the grunt work, right, with this online tool, and then you're able to let your experts actually be experts, as opposed to just be grinding through routine stuff. So it really is an assister as much as it is, I don't know if that's a word, but it's more of an assister than it necessarily is an automator. Right? It goes through, it does this stuff, and then, because ultimately what you're trying to do is get through these images accurately and quickly, right? Because it's time and money, but if you don't do good science, it doesn't mean anything, right? And so when you put that QA spin on it, if you will, it provides a whole new level of value for your team.
Yeah, and that's a great point, Tim. And what we say here at the Jackson Lab is there's really no such thing as automatic image analysis. We call it semi-automatic, because you do want that expert QA across it, and you want the automatic algorithm to facilitate that QA, not be some kind of black box that you're just looking at, and right. This approach works very well. So the other measurements that are being made are counting the number of vertebrae as well as the number of ribs, and they use a very similar approach to the digit, and here are examples of those. And so there are far more measurements to be made on these DICOM images, but this right here alone will take away approximately, we figured something like more than an hour and a half from a data set per knockout line. So that's reduced the workflow tremendously for whoever the phenotyper is. And then, as stated earlier, it's the other real value is the way it's outputted can directly flow right into the data coordination center data tool that picks up the data every night and analyzes it and then presents it to the public. So it's always an ongoing process. This approach allows us to feed directly into that system. So it's very valuable from that. So, and then the second case study is for optical coherence tomography, and this is a method that uses interferometry on live mice to get contrast across tissue, in this case through the eye and through the retina. And it uses a reference beam alongside the beam that goes through the eye, and it achieves contrast by looking at the difference in interferometry between the two beams, and generates an image which Tim mentioned the other day looks a lot like an ultrasound, which it does. And what you're seeing in the running movie is a lateral movie through the retina. So you're looking basically at a cross-section of the retina, but it was imaged and then imaged along the retina, and then a movie made of basically a lateral projection through the eye, through the retina. And you can see that there are layers in there, and those are all the different layers of the retina, which I'm not going to go into, but the point is that the layers and their width and the spacings between them have relevance to different eye diseases, and so being able to rapidly measure those would be a very good high throughput phenotype for assessing retinal problems. And so we sent these data to ImageIQ, and they came up with an IQ bot for it that automatically marks these things and will make the measurements between the layers automatically and assess those and identify the outliers. OCT is an expensive piece of equipment, but the amount of information you get is much greater than the other standard ways of looking at the eye. And it does require a little more expertise to run, but if we could provide automatic algorithms to make the measurements and to assess the samples, then it's a very useful technique that could be adopted by centers which don't have on-board expert eye phenotypers. And to be honest, there aren't that many around in the world, and we're very lucky here at the Jackson Lab to have a lab, the Patsy Nishina Lab, which is pioneering this with Dr. Mark Krebs, who's an associate research scientist in her lab. And they would really love to see this technique be adopted across all of the IMPC and common phenotyping pipelines. So it's in discussion now, but having this kind of approach is what's going to make it feasible.
Yeah, Jim, it's kind of like, we talked about the ultrasound thing, but you look at even today in diagnostics, in clinical care, right? Your quality of your ultrasound is dependent on your tech and dependent on who knows how to read it, and usually your technologist works very closely with your radiologist, right, in that space.
That's right. Yep.
And that's why, if we can take these kinds of tools and make them available in that online image analysis, what it does is it enables you to think about these tests, because I can't fathom, we talked to Mark about this, right, and he's like, look, there's all this data in here, but I can't fathom how could we roll it out across 18 sites, and how can we get everybody doing this in a way that is consistent and reproducible. And I think that's where, again, we talk about the power of online tools. That's where you can start thinking about those enabling this kind, for the science minds to start thinking about, I can do this. That's the exciting part, right? That's what makes it all sing.
Right, that's right. And then the next slide discusses our fundus imaging. So this is the traditional way of phenotyping the mouse eye, and that is to just look through the eye and look at the back of the eye and take an image of it, or note without an image, but just to have the phenotyper note abnormalities. And this is it's a time-consuming test, and the images are very hard to read for all sorts of reasons. The eye is curved. Getting the fundoscope up to the animal is physically challenging. And in the case of the Knockout Mouse project, the strain of mouse that we're using, which is the C57 Black 6, there is an inherent lesion that's associated with the retina due to a mutation in a gene called RD8. It's a background mutation. It's been there for a long time. It's difficult to train phenotypers to recognize lesions that would be associated with the knocked out gene over that background of the inherent lesions. A good eye phenotyper can spot the difference quite easily. It takes an eye a while to pick out any kind of pattern in anything, in any kind of work that you're doing. Same thing here. So we asked ImageIQ if they could come up with an algorithm that could automatically identify these lesions and score them based on location and size. And in that, by developing something that works that way, this will bring the abilities of identifying true lesions, or not true lesions, identifying lesions associated with the knockout over the background lesion to all the other centers. Along those lines, we had an eye phenotyping workshop here at the Jackson Lab this summer for all the IMPC and comp centers, and it was really informative for everyone to see what experts would identify as a background lesion versus an actual knockout lesion. And I think it brought a lot of homogeneity to the phenotyping. It also really brought to the forefront the need for some kind of automatic analysis that's more, as unbiased as can be, and allow for a more rapid throughput. And before we go to the next slide, I want to say that not every mouse has its eye imaged. It's only the ones that are identified by the phenotyper as an outlier. That's to increase throughput because it's difficult to take the image, and also it's difficult to read the image. But it would probably be much more helpful if we could image one eye from every mouse, and if we had a good image analysis algorithm to annotate, then that would make it a really worthwhile effort to bring more value to it. So, so on the next slide, it shows that the green in there is the identifying of the lesions, with the vasculature and optic nerves being segmented in the blue here. So the challenges on reading these images, as I said earlier, is the curvature of the eye leads to a lot of uneven shadowing. Also, there's no standardized coordinate system for looking in the eye at the moment. There is for fixed eyes, etc. When you have a live mouse in your hand, there's no inherent, really rapid coordinate system to allow you to know where you're taking the image in relation to points on the eye, other than where the optic nerve is, but you don't know what your angle is to that. So there's some variability in presentation of the image from image to image in terms of what areas of the eye are showing. And then the vasculature and the optic nerve can also create what looks like lesions in areas along here. And so what this system does is identify the lesions based on coloring, shape, and size, and can provide you with an analysis of the number of lesions above what's considered background for a black 6, as well as sizes. So the RD8 mutation creates lesions that are typically smaller than most other ones. So here's a method that very rapidly can allow you to assess the extent of the lesions, the areas of the eye it's in, their area, and I believe that you can turn these on and off and look at the underlying image itself to assess in your own QA whether that's a real lesion or not. So again, it provides you with expert phenotype QA after the analysis program to vet it. So in summary, the automatic analysis of images is really valuable at increasing our throughput. It's a necessary aspect of managing images, and it removes, or has potential to remove, bias. So any manual annotation will always have that inherent user bias. It takes the weight off the experts, and it also allows institutions which don't have on-site expertise the ability to annotate these data with essentially the input of an expert through the algorithm. Reduces the need for manpower, and it allows for access, like we mentioned earlier, for any center that wants to utilize these tools. It doesn't have to be on board in their institution. It doesn't have to be on their computer or on their hard drive. So it's available to all of the IMPC sites. And so it's been very helpful for us.
Very cool. Well, Jim, I think this is Tim. I think the next slide might be something that I'm going to spend a little bit of talking about, and literally 5 more minutes as we kind of get to the end. So just probably as a reminder, I'm sure if you guys have any questions, type them in. We'd be happy to talk to you about them or hear what you're thinking. One of the things that fascinates me when we get engaged with someone like the Jackson Lab and Jim's vision of how groups across the world can work together is, well, how do we solve those problems, and really, what can the technology do for you, right? I mean, ultimately, it used to have this saying, people used to ask me all the time, what language is your technology written in? They're like, look, you shouldn't care if it's written in crayons, right? As long as it solves your problem. And so, the technology that we've seen working really well here, both now with Image Quantify as well as like the Omero solution that you're all using, is really that you've got the ability for cloud-based data management. You've got the ability to analyze these images in the analysis is automated. You give it an image, it automates it, and then you decide what to do with it. Do you QA it? Do you triage it? Do you prioritize it? How do you deal with that? It's in the secure cloud, so nobody's standing up disk drives and all that kind of craziness, right? You get, as Jim talked about, you actually get data. You get real hardcore. I mean, they're spreadsheets. It sounds cooler when you say database, but they're spreadsheets that you can use for this. But you get those overlay images, and Jim, like you just talked about 2 slides ago, right? You can see those overlays. You can turn them on and off so that you can look at the underlying original data and kind of have that augmented experience, which works really, really well. And you get references and you have all the other stuff. But you can see that we bring that technology to bear. But the real question is, why now? So we sit here in 2015 and say, well, why in the heck are we doing this now? Why are we having a webinar about online imaging tools, right? And I think it's important to realize that there are technology enablers that all of you, as you're continuing to do your preclinical work and your imaging, need to keep an eye on, like, a little bit, and know, because it will help change the game in what you do, whether it's collaboration or analysis or storage or any access, any of that stuff. And I think this is kind of fascinating. I just, a couple weeks ago, I finally got around to watching the movie called Imitation Game. If you've ever seen it, it's a good computer science-y movie. But I don't know, Jim, have you seen it? Have you seen Imitation Game?
Yeah, it's very good.
It's a good one, right? It's Alan Turing, and I'm not sure exactly how hyper-historically accurate it is, but it's around the concept of him working on breaking the Enigma machine during the war, the German Enigma machine during the war. Put all the words in the right order, it makes more sense for everybody. And one of the things, you looked at his machine, the early stage of the computer, and you wouldn't be giving it 5 Faxitron images and asking it to find the digits and vertebrae, right? I mean, it was having a hard time doing anything, right? So at that point in time, there wasn't the technology available. But as certain technologies start to converge, innovation happens, and then it enables a new way of thinking. So, what we see as the enablers for online tools for all of you is that, in a lot of cases, algorithms have become more mature. Our folks who do core research on analysis algorithms in the community and the ability to analyze images. Jim, you said it almost as naturally as you talk about going to pump gas, right? They're DICOM images. Well, 15 years ago, DICOM didn't exist, right? It was a gleam in a couple of guys' eyes who I've had the good fortune of actually knowing and working with. But we didn't even have a standard for moving the images around, and that's plumbing. I mean, that's not even anything. So the algorithms to define to move the images around are now available. We have computers that can handle them. We can think about an X-ray, or we can think about an OCT image, and we can talk about storing gigabytes or terabytes worth of image data, and it doesn't freak anybody out. We're not talking about robotic tape arms or anything like this. And we can now move them around, right? Internet speed, I mean, heck, you can download movies from at home or on your phone. I just saw, for those of you in time, I'm not sure if we're recording this or not, but in time, while we're recording, it's the day after the 2015 fall Apple announcement. And they talked about the new LTE Plus, where you're going to get 300 gigabit of download over a cellular connection. I mean, it's crazy. I don't think we get 300 gigabit at the office. I mean, we're just going to give everybody cell connection. So we've got internet speed that's really helping people move these images around. We've got this concept of cloud computing, and let's not make that too fancy, but the bottom line is cloud computing is someone else's computer in a data center that you don't have to worry about. You can have more of it or less of it, and you pay as you use it. Right? It's not any more complicated than that, but you know, you all don't have to stand up server farms anymore. You don't have to get a bunch of storage enabled. There's all sorts of those kinds of things. And you bring security technologies together with that, and I would say most of us probably do banking online, and you know, we probably manage our 401k online, and certainly you can order a pizza online. So there's all sorts of really good secure technologies, and you get the blips where somebody hacks some site, and that'll happen. Yeah, Jim, I know in rehearsal we were talking about this. You know, I used to be an analyst for the Navy, and we had somebody come in to assess our security of our system, and he said, you know, I can't do his accent, but it was good, strong U.S. Navy accent, if you will. And he said, look, the most secure machine is the one that's turned off and locked in a closet. He goes, other than anything beyond that, you're going to have some security risk. But in general, security, we are doing banking online. We are doing a lot of transactions over the internet, and everyone's connected. And Jim, you said that before. You know, you don't have to worry about deploying this on a bunch of machines. You put all those things together, and it enables online imaging tools, right? And if you don't have all of those, something falls down. But once you kind of get all 6 of those things in place, you have a foundation where, you know, Jim, people like you and your team can come and say, I want to try this. I want to have this be accessible to our group. So it's kind of a fun enabler. So when you put all those together, you get some neat things. You get something that's globally accessible. You get something that's scalable, which is amazing. I mean, we will never, as big as the comp project is, we will never push as much data through the Amazon cloud or IBM's cloud as everybody else, right? So we've got conceptually infinite scalability. I was teasing Jim the other day. I said, you know, if in theory, if Jackson Lab acquired all 10,000 of their images for 5 years and said, you know, I'd just like to get them all analyzed, and the analysis algorithm takes 3 minutes to run, we could put those on 10,000 virtual computers and have them all done in 3 minutes. I mean, it's somewhat of a theoretical argument, but it's not too far off from reality, right? It's kind of weird. It's kind of weird. I don't know if that would freak anybody out, Jim. I mean, be kind of bizarre to do.
As long as they got the data rapidly.
Yeah. That's true. That's true. The other nice part about this stuff is it's deployable instantly, right? And you don't have any hardware to deal with. And these are benefits that, you know, people like to talk about them, and they show up on TV shows where people go, oh, the cloud and all this kind of stuff, and they're marketing and all that stuff. But at the core, it's a tool for all of you, and that's kind of the key thing. So. And I think that ultimately enables better science. It speeds your time to analysis, it produces those objective results, it reduces cost, and it creates a real standard of practice. And you look at the challenge, you know, I love the Hadron Collider analogy, Jim, right? I mean, because what you guys are doing, you're not really colliding mice into each other, but I mean, it's on the order of that. It's a big international consortium of people trying to solve a common problem with resources and expertise in a lot of ways, and you got to get the standard of practices right, and your SOPs are a foundation for that, and this just augments that, right?
Right.
You know, and then, you know, finally, it's kind of simple. So it brings it kind of all together. So I think, you know, with that, I mean, I like to talk about the technology and stuff. Jim is obviously the biology expert and the process expert and knows about all the things that are going on here. I thought, you know, Joanne, we'll turn it back to you to maybe take a few questions from the audience and see if anybody's got anything they'd like us to talk about as we wrap up.
Oh, thank you so much, Tim, and thank you, Jim. This is really fascinating. Yeah, we do have some questions. Would you talk a bit about how researchers can get access to this data?
Oh, yes. So the data portal is at the IMPC, and the website is www.mousephenottype, all one word, mousephenottype.org, and it's the public site, and there is a search box by gene or phenotype or even protocol in the right side of their homepage, and it's pretty intuitive how to use the site. So go there, mousephenotype.org.
Hey Jim, how often is that data updated? Because you mentioned you guys do daily uploads and stuff like that. How often is that refreshed? If somebody searches it today, when is it? 3 months, 6 months?
I don't know, Tim. I don't know. Because once we upload the data, it does take a while for it to go through QC at that site. Sure. It's updated frequently. Not every 3 months, but I don't know the exact time. But I believe there's also a place where you can ask for updates for your genes of interest. I think you can create a search that will keep you updated. If it's not operating at the moment, I know we had talked about putting that in, and so that possibility will exist soon if it's not there already.
Beautiful. So for Jim, how much training do fundus reviewers usually receive before they are qualified to read those images?
Well, here at the lab, I would say several months of hundreds of animals. I can't speak for the other centers. We have a strong group of eye researchers here, and we have lots of models for which we can use for training. So, which is why analyzing automatic analysis of fundus images is really important, because we're a little bit unique in having the real strong expertise here. So it really highlights the value of having an unbiased method that can rapidly score or show you the potential outliers.
Thank you. Tim, would you talk about the process of customizing those iBots?
Oh yeah, sure. That's a great question. You know, it's the bottom line is usually what happens is if somebody comes to us with a problem and say, hey, I need a new IQ bot that does X, Y, or Z, does some kind of analysis like you've seen here. The process is relatively, I don't know what's the best word, predictable, right? What we do is we like to get some sample images, we go through some proof of concept to see if what you're even asking for is possible, right? Because, you know, some of the stuff, as Jim pointed out, some of the stuff you can't do with software. This is not a magic thing, right? So we see what's possible. We go through that proof of concept, and then if the proof of concept works out, what we'd like to do is get a cohort of images and go through a pretty strong human observer validation process. I referenced that where my eyes were rolling back in my head before, right? So that's what happens. We get a number of people to go through and make the same quantitative measurements on the images that the software is going to make. And not everybody does that. A lot of times we see people grading images like on a scale of 1 to 5, and it's kind of that qualitative sort of assessment. We will go through and make the same quantitative metrics, and then we do, you know, we get the stats folks involved, and we actually go through that validation. And so that whole process, depending on how close of a bot we have, we call them cousin bots, right? We have a bot that does one thing, and if we just have a little cousin, that can be as quickly as a couple of weeks. It might be more like a couple of months, depending on the conflict, the way it works. But then once it's done, it's validated and available.
Great. And so is this something that you're doing, is there a similar model that you're doing with other collaborators? And this is a huge collaboration with Jackson Labs and all those sites, but are there other collaborations that you're involved with now?
There are, yeah, there are. You know, it's amazing, and I think this is what makes, for the folks at Image IQ at least, right, I can only speak for us, but which makes our days always exciting. The number of kinds of images, the imaging modalities that are out there, the types of things people are trying to measure are really exciting. So we're doing this with all sorts of stuff. Obviously, front and center for a lot of it is Jim's work with the COMP and IMPC projects in Jackson Lab. But yeah, we're working with other universities, other, you know, very honestly, the preclinical research departments in commercial groups, right, who are trying to figure out what their next product line is going to be and what the next technology foundation for them is going to be. So yeah, it's kind of fun. We call it, you know, we got a little catalog of IQ bots, which is kind of nice.
Great. So as I'm hearing your presentation, I'm wondering, have you also seen sort of hackathons begin to develop? I'll just share with you a bit. A couple of years ago, we tried to put together a hackathon, and what we found was that we had no trouble finding technology people, software developers, app developers, and so on, who were excited about working with biological data. But we had a very difficult time getting people from the pharma industry to describe their problems in order order to create an appropriate challenge to do a hackathon. You know, we wanted to do something very specific to our world. We didn't think we needed to sponsor the development of menu or parking options, which is what many hackathons are doing. But, I just wondered, have you seen anyone start to kind of pull around this massive data that's now available publicly, and to start to interrogate and look to develop other layers that could be helpful to researchers?
Was that addressed at me?
Whoever. Yeah, go ahead, Tim.
Yeah, well, I can address that. Here at the Jackson Lab, we have several PIs who are going through all of the phenotype data and looking for relationships between them in different combinations of pairings, basically to try to identify relationships between phenotypes and genes that are not obvious. So, and that's kind of a hackathon approach, and it's really it's exactly the kind of approach that is needed for looking at these data sets. It can identify relationships between genes that were not previously known or even thought about, and so it requires a lot of creative analysis of what kinds of approaches to take in terms of grouping the data to compare it to. So, and that's where the future is in terms of the data analysis at this point. So an awful lot of effort has been put into building the infrastructure for creating the mice, the knockouts, creating the mice, building the phenotyping pipelines, getting them vetted, getting them to be QC'd, and creating strong, valid data, and getting those data into a place where you can get to it, which that's really critical, is how can I actually readily get to these data. And now we're just starting to turn to the data in a big way and say, well, what are all the different ways that we can manipulate these data to look at unforeseen or creative ways of extracting more information from the data. And so that's really the exciting challenge for the next couple of years. And Alyssa Chesler and Vivek Kumar here at the lab have just started that and presented their work to the IMPC at a recent meeting, and other groups are now starting to get behind them or work with them on doing that. So that's the future, exactly that, and it's sort of a hackathon.
Mm-hmm. Mm-hmm. That's exciting. You know, I don't think you noted anywhere, when did this all get started? How long have you been at it?
For the phenotyping, so there are three parts of this round of funding. There's funding for production, which is to take the knockout ES cells and turn them into live animals on the shelf. The second part is for building the phenotyping pipelines, and the third part is for constructing the data coordination center. And we're coming up on the 5th year of funding, and so it started approximately 5 years ago for all those separate grants. And we're waiting for the RFA for the next round of funding, which should be coming out anytime now. But prior to the actual building of the mice and the pipelines, there was a Comp 1 grant, which was for making the knockout constructs and the libraries of embryonic stem cells for making the knockouts. And so that was ended just prior to the start of the production and phenotyping pipelines. So it's been approximately 10 years in the making, from concept to collecting real data and then analyzing it.
Well, this is terrific. I think we're just about ready to wind up. Would each of you like to make any kind of final statement before we say goodbye? Let's invite you, Jim, first.
Thanks for inviting me. I like talking about this, and it's important. One of the missions here at the Jackson Lab, as well as across the IMPC and the Comp, is for public outreach and to let the public know where their tax dollars are going, and it's going to something that has a lot of value for translational and clinical medicine in the future.
Beautiful. And Tim, any concluding remarks?
I think, yeah, I think it's the same thing. Is thanks to the BioPharm team, Joanne, you personally, who've really helped pull this together to let us share some of what we're trying to do to help all the cool work that's going on. I think it's great. You know, we think there's a lot of power in imaging and the work that Jim and the IMPC are doing, and we're just happy to be, you know, part of helping make that happen.
Fantastic. Well, thank you, everyone. Thanks for everyone for joining us. There will be a recording on our website within a couple of